Last time we ended up talking about ribosomes, and we were talking about all the subunits, all of the tRNAs, the ribosomal RNAs, the tRNA synthetases that actually do the work of pairing up the appropriate tRNA with its anticodon to the amino acid. So we got all of basically the raw materials talked about. Now we actually need to talk about actually polymerizing this new protein chain. So here's the crystal structure of the ribosome. Very elaborate, very amazing, but we're going to completely ignore the details. And now we're just going to talk about the ribosome as these two blobs. There's a big ribosomal subunit, and there's a small ribosomal subunit. And I know this slide is busy, but we're going to walk through it, so don't worry about that. OK. There's a small subunit that's sometimes called the 30S or the 40S. So the 30 is in prokaryotes, the 40 is in eukaryotes. And the S stands for sedimentation coefficient. It's basically how heavy the molecule is. Like the early biochemistry analysis of this, you put them in these gradients and you saw how dense they were. So these are density ratings, kind of just to give you a general idea of how big they are. We actually know now how many proteins are there, what the size of the proteins are, how many RNAs there, the length of the RNAs. So we have much more elaborate, detailed ways about talking about their size now, but these, for kind of historical reasons, have stuck around. All I want you to know is that there's a small and a large. Okay, so the small is sometimes referred to as the 30 or 40S, the large is the 50 or 60S. These are two separate domains, and in the cytoplasm, these are floating separate from each other. Okay? So here is our newly made mRNA. It's already been spliced and edited, exported out into the cytoplasm from the nucleus, and now it's ready to be translated. Okay? So it's going to come out here. And the first thing that happens is the small subunit with some associated initiation factors. And I'm glad that the text makes these generic, because I'm not so interested that you know specifically. But it does take accessory proteins to get that first ribosomal subunit stuck to the mRNA. Okay? So there's a couple of proteins that help it to find its appropriate mRNA. And the small ribosomal subunit is the first thing to sit down. So it sits down here, and it sits down on the region called the 5' UTR, the 5' untranslated region. This is a part of the mRNA, but it's not going to actually be made into protein. Okay? This is the AUG. That's the first start codon. Everything upstream of the AUG, that is everything to the 5' side, is not going to get made into amino acid sequence. It's there for regulation purposes and there to get the whole thing set up. So it's the UTR, 5' UTR. In prokaryotes, they have a specific sequence called the Shine-Dalgarno sequence. It's named after the two guys that discovered it. That's the sequence that the ribosome is specific to sitting down on. Okay? So the small ribosomal subunit will recognize that Shine-Dalgarno sequence. And then the first tRNA is going to sit down. Okay, so first the 30S or the small subunit, then the tRNA, and it's going to bind to its appropriate anticodon. Okay, the start is almost always AUG, and so the first tRNA's anticodon is UAC, right? The complement of that to sit down. All right. Once we've got those two sitting down, then the big subunit can come in. The big subunit is going to be the one that's actually going to do our polymerization. But in order to kind of get everything lined up properly, the small subunit is the one that's first. Yeah?
Do we need to know the name of that thing it sits down on?
The Shine-Dalgarno? Yeah. Okay. That's in prokaryotes. Eukaryotes have, it's not the same sequence, but a similar thing. There's a specific sequence that the ribosome sticks to. Right? So here is the first codon. You can often have alternative start sites. So there might be actually several AUGs on the mRNA. And depending on the cell type and depending on the environment, you might actually start at a different place. So you could actually have a couple of possible places. And you just have to experimentally figure out which one's the right one. So you can read the sequence, you can find the AUG. That might not be your start. It might be an AUG that's 10 base pairs down or so. So there's alternative places to sit down. Now, once it picks the appropriate one, the big ribosomal subunit sits down, and it sits down with that first tRNA that's got a methionine on it. So tRNA, it's got methionine. It's going to sit down in the middle of 3 sites. So there are 3 tRNA sites inside the big ribosomal subunit. They're labeled, we call them the E site, that's this one. The P site, that's where the first tRNA sits. And then the A site, okay? Now we have both subunits together, and so instead of calling them the 50 or the 30 or the 40 and the 60, we're now into the 70, or I guess it would be 100 in the eukaryotes, okay? So everything's together now. Now we're actually ready to start reading the mRNA sequence and polymerizing our amino acid. Okay, so it's the 60, the large guy that's got A site stands for amino acid site, or you can think of it as the addition site, where the next amino acid is going to be added. So as this thing starts reading the mRNA, the A site is where the next codon is going to be read, okay? The P site stands for polymerization or polypeptide. That's where your long growing protein chain is going to be, okay? So right now it's not growing yet, it's just got the single methionine, but that's where your long polymerizing protein is going to be sticking. The last spot is the E site. This is the exit site. That's where your used up tRNA is going to leave once it's done its function. So A, P, and E. Did you need something?
Just the E. What is it?
E, exit. When you've used up that tRNA, that's where it dumps out. Yeah.
Just to clarify, is the 5' UTR different? Is apart from the Shine-Delgado?
The Shine-Delgado is a specific sequence in the 5' UTR. So everything, AUG, that actually codes for methionine, so that would be part of the coding region. Everything, every nucleotide to the 5' side of it is called UTR. The Shine-Delgado is the actual sequence that the ribosome sits down on. Okay. All right, so then we start elongating. Where the ribosome is sitting determines the frame. So where that first AUG was, the ribosome sits down on that, the tRNA sits down on that, and the next 3 nucleotides is the next codon. This is the frame, right? I'm sitting in this frame because I've decided that that AUG is the first codon, and so after that, we're going to read each next frame is just a series of 3 codons, 3 nucleotides and a codon, right? So this is the A site, the amino acid addition site. You know, that's just determined because that's where the ribosome is sitting down, right? If the AUG was not there, if it was AUG here, I would have shifted over a bit and I'd be reading a different frame, right? So the amino acid site, another tRNA comes in, its appropriate anticodon sits down here. This example is showing you really nice base pairing, but remember that 3rd position could be wobbling. That might not be exact. This one codes for serine, so that's the next amino acid in our growing chain. All right. Yeah?
I'm trying to understand the A site has to do with everything again. Like the first sentence, the next codon determines which tRNA will be the A site. Can you just
Will
explain
bind
that?
to the A site. Right. So these little cavities here are the 3 sites in the ribosome.
Okay.
Okay. And this guy sits down on the AUG. That means that that is where the ribosome sits, right? That's defining where it can sit down. Okay. Then this A site, this cavity right here, is sitting on 3 nucleotides, right? So it's where it's sitting is determining what the next 3 nucleotides we read are. Okay. hopefully it's self-explanatory, but it's the structure and it's the size that determines where it is.
Okay.
Right. So that allows a tRNA to come in, and that tRNA fits in the pocket and lines up with its appropriate anticodon. And that's what tells you, okay, yeah, that's the next amino acid that needs to come in. Right. So it's not, I'm saying that because it's not reading it, right? It's not reading the next 3 nucleotides. It's just saying whatever the next 3 nucleotides are, that's the tRNA that needs to come in. So, I mean, the ribosome is an amazing molecule, but it's not reading anything, right? It's just structurally hovering over a site. Okay, so in order to polymerize this growing chain then, what I've got to do is break the covalent bond that's currently holding the methionine to its tRNA. Okay, so this is the big subunit of the ribosome, clips that covalent bond, and then it reforms the covalent bond through a peptide bond. So it takes that methionine, sticks it on the serine, which is the next one, and then forms the peptide bond. That's the chemistry that the ribosome is doing. Right? Breaks that covalent bond, reforms a covalent bond in the form of the peptide bond, and polymerizes these together. So the growing chain now is always going to be attached to the tRNA that brought the most recent amino acid in. Okay? So the newest amino acid comes in, and whatever's been polymerized so far is going to move over and jump onto that new site. Okay? So right now we only have 2 amino acids, but this could be 100 amino acids, and the 101, the entire thing is going to move on to the next tRNA. Okay? So it always remains covalently bonded. All right. Now the whole thing is going to shift over. So this is just peptide bond is formed. This is called peptidyl transferase. It's just the definition of the enzymatic function that it's doing. And I put this up because it's actually the RNA pieces in the ribosome that are doing the chemistry. Most of the time we think of enzymes as being proteins. Proteins are the ones breaking and reforming bonds in their active sites. In the ribosome, the active site actually has RNA, and it's the nucleotides of the RNA that are providing the catalytic domains for breaking the bonds and reforming these. So this is one of the unique places where you actually see RNA as an enzyme. The RNA is doing the chemistry. So once we put on that second amino acid, the ribosome shifts over now, and it puts the polypeptide, the growing protein, in the P site. So here's the tRNA left over from the methionine. It's now in the exit site. And the ribosome now is going to break the hydrogen bonds that hold that guy and release it. So it shifts over. The methionine tRNA moves. Hydrogen bonds break. And that tRNA floats off into the cytoplasm. And it's going to get recycled. That's still a good functional tRNA. It can get grabbed by another tRNA synthetase and another methionine stuck on it, and it can get reused again. Okay. Now I've got my growing peptide in the P site, and I've opened up a new empty site. And the cycle just repeats, right? Now these 3 nucleotides are in the A site. That's going to allow the next tRNA to come in, bringing its next amino acid in, and then I will shift these 2 amino acids onto the next one, and we just keep shifting over. So you just shift over 3 nucleotides, put in a new one, shift over. We're just constantly recycling 3 nucleotides at a time. We got it? Questions on this?
So the tRNA attaches after the codon gets into the empty A site?
Yeah.
Okay.
All right, so you just keep doing that for however long your protein is. And that's determined by how long your mRNA is, right? So I just keep moving down the mRNA. This could be for hundreds of nucleotides. So here's the P site. This is the last tRNA that came in bringing a lysine, and then the whole rest of that whole amino acid is still attached. Okay? Then we come to one of the stop codons. This is UAG. They're multiple stops, right? There's 3 different stops. UAG is the one we're seeing here. That's sometimes called the amber stop, named after one of the guys that helped discover it. And then when they found multiple stops, they kind of kept the color theme going on. So amber was named after the guy. He was a German guy, and his last name meant amber, so they named it amber. Then they found the next one, UAA, and they named that ochre. And then they found a third one, UGA, and they named that one opal. Those second 2 aren't named after anybody. They're just kind of, I don't know, inside jokes, I guess. I only put them up there. I don't care that you memorize them. You don't need to know. But sometimes you'll read in the literature and it'll say the opal stop or the amber stop or something like that. You should just know that it's differentiating between the possible stop codons. As long as you know the sequence of the 3 stops, you don't need to know their nicknames, right? When a stop codon enters that A site, something unique happens. There's no tRNAs that correspond to any of the stop codons. Okay? So there's no amino acid that's going to be brought in. What binds to that is actually a protein called the release factor. Okay? So it binds in here, and it just creates mayhem. It just breaks everything apart. That's its job. So I've already got my whole protein made. All I've got to do is just basically disassemble the whole thing. So it breaks multiple bonds here. Breaks the tRNA polypeptide bond first. So that whole polypeptide chain, the last guy that was on there was the lysine. So the release factor catalyzes the breaking of that bond, and it first just releases your finished protein. Okay? And then it's going to disassemble the whole thing, but I'll take your question, yeah.
So does the protein go through like secondary and tertiary conformation while it's getting added?
Yeah.
Right?
Yeah.
And then at the end, it can be taken to...
Yeah, we'll talk about where it goes, but you're right. I mean, once you get a couple of amino acids, it's already outside of the context of the whole ribosomal thing, and so it's starting to take on its secondary conformations. Yeah. So if it's got sequence to code for a alpha helix, it'll start coiling into an alpha helix. It'll start forming beta sheets. And at that point, chaperonin proteins may even start coming in and folding it into tertiary structure as well. So yeah, that's all going on right as it's being made. So it never exists as just a floppy noodle. Yeah, it's always adopting conformation. So yeah. Okay, so the release factor releases from the tRNA. So that releases the peptide. Then it also releases so it released the protein from the tRNA, and then it breaks the hydrogen bonds here, releases the tRNA from your mRNA. And once that happens, the whole thing then just breaks apart. That tRNA is kind of fundamental in holding the 2 ribosomal subunits together. So the ribosome falls apart at that point. And those units could then recycle back, grab another mRNA, and start making something again. But yeah.
I was just wondering, you said the release factor does something to the peptide chain?
It breaks the bond. That peptide, that last amino acid was bound to the tRNA by a covalent bond. It breaks that bond and releases the peptide. And then it breaks the hydrogen bonds of the tRNA with the mRNA and releases the tRNA. Once those 2 things have happened, the whole apparatus just falls off the mRNA.
Okay, I thought I heard you say something about how it adds something to the polypeptide chain as well.
No, it doesn't add anything.
Okay.
It just cuts off that tRNA. Yeah, maybe I misspoke. We've talked so far as this just being in the context of one of them going along the mRNA strand. You can actually have multiple ribosomes reading the same mRNA strand at the same time, because so here's the first AUG. This is not showing you the 5' UTR, but there would be 5' UTR out there. So, a ribosome could sit down on the start site and start moving down. So the first one has moved all the way down here. It's got a really long peptide. Well, as soon as it clears that first area, and the Shine-Delgarno or whatever the 5' UTR sequence for attaching the ribosome, once that's cleared, another ribosome can come grab onto it. So you can have multiple ribosomes chasing along the same mRNA. So this is called a polysome, right? Now, in this illustration, I've got 1, 2, 3, 4, 5 ribosomes making 5 proteins off of the same template. Right? And as soon as this one falls off, everyone shifts down, I could put a 6th one down. And I could just start making this. So every single mRNA is going to allow for the making of multiple, many proteins. And as long as that mRNA is around in the cell, you're probably expressing the protein. Now, not always, but once you start getting an active mRNA, it could be made multiple times. Okay. Yeah?
Are there like specific ribosomal subunits that are made for a certain mRNA, or is it just all generic for?
Are there certain ribosomal subunits made for certain mRNA?
Yeah.
Well, there's 2 in every eukaryotic cell. There's going to be a mitochondrial. So the mitochondrial genome has a slightly different genetic code. So it has to have some of its own tRNAs, some of its own tRNA synthetases, its own ribosomal proteins. So the mitochondria has a slightly different ribosome than the mRNAs made off the nucleus, the ribosomes that are hanging out in the cytoplasm or on the rough ER. So yeah, those would be 2 different ones. If you're in a plant, the chloroplasts are going to have a different ribosomal apparatus mechanism. So that's true. But in terms of like every mRNA coming out of the nucleus, it's the exact same ribosome that does it for every one of them. So that's why, so the ribosome is generic, right? It's not reading anything. It's just whatever mRNAs are getting spit out of the nucleus, if it can bind to them, it'll start making that protein. So there's no specificity there. All right. The example I've been showing you as well has just had a peptide chain. This new protein has just been being dumped out into the cytoplasm. And a lot of proteins get made that way. There are ribosomal subunits that are just floating around in the cytoplasm. They grab an mRNA, and you could just make that protein right there in the cytoplasm. Now, a lot of proteins, however, have to be specifically made at the ER, at the rough ER. And the mRNA actually tells the cell that I'm the kind of protein that needs to be made in the in the ER. Okay, so here's a ribosome sitting down on an mRNA. Okay, here's my 5' end. It's sat down, and so far it's made a little bit of protein. That protein actually will code for certain amino acids that are like, I don't know, they're like zip codes for the cell, basically. If there's a specific amino acid sequence called the signal sequence, that signal sequence is bound by a certain other protein in the cell. So the first thing that gets made off of that mRNA is the signal sequence of amino acids, and a signal recognition particle will bind to it, right? So if the mRNA makes that signal sequence, it's basically saying, I'm the kind of thing that needs to go to the ER, and the signal recognition particle says, oh, here's an amino acid sequence telling me that this needs to be in the ER, and it will bring it to the ER membrane. Okay, so here's the ER membrane. There's this little docking protein, and that guy binds up specifically with this signal recognition particle protein. Okay, so the signal peptide is actually amino acid sequence in the protein. It's the zip code, right? This is the mailman, right? Recognizes that, oh, that's the zip code that needs to take it to the ER membrane, and then it sits down here, I don't know, on the mailbox, right? Recognizes that's where it's supposed to be. Then, as the protein is continued to be made, as it gets made, it actually goes through a little channel and gets pushed into the ER. So some of your proteins are just getting dumped right out from the ribosome into the cytoplasm, but ones that have a signal peptide are going to get forced into the lumen of the ER. So the signal recognition particle docks. It actually takes the signal peptide and swaps it with another membrane-bound protein there, and that holds it there. And then that signal recognition particle could float off. The rest of the protein gets made, dumps into the ER, and now I've got an ER-specific protein. Then when it's done, everything dismantles, right? Stop codon, that protein, the terminator protein comes in, sits down, breaks everything apart, and I've got the protein now inside the ER. And then, ER can modify it, and it can be passed on to the Golgi, and I can do all those protein modification things of it necessarily. The bulk of your proteins actually get made on the ribosome that are on the ER. So this is rough ER. What actually makes ER rough is the docking proteins that are there, because the actual ribosomes stick but then get released. So if you take a picture of rough ER and you see all the ribosomes, they're not there permanently. They're only there because a recognition particle told them to bring them there. And once the protein gets made, they're going to fall off, and another ribosome with another mRNA is going to come attach. So that's a really dynamic membrane there. It's not static. Those guys aren't just sitting there. The ribosomes aren't just sitting there waiting for a protein or an mRNA to come to them. They're actually actively recruited by this mechanism. So, all right. Questions on translation? This is where I'm going to stop. I'm not going to go through the whole protein modifications and stuff like that. You've probably seen that before in Cell. So, yeah.
Just to clarify with this, once that whole complex, the ribosome is docked, does the sequence always get shoved into the ER after
Yeah.
that? And that's why it
Yeah.
needs to be docked there.
Yeah, I mean, that's the whole purpose, is to get it shoved into the ER, right? Yeah. There's nothing else, at least I don't know of anything else interesting about the membrane of the ER. I don't think any interesting modifications are going on there. It's just places to get inside. Like all of the interesting modifications and stuff are going on in the lumen of the ER. Yeah. So then it's going to clip off that signal recognition. That signal peptide gets cleaved off as well. And it gets left there, and it'll be released eventually. But once the zip code has done its job of getting it properly to the ER, then it's cleaved off. There's multiple signal sequences on proteins as well. So this is just the first in many zip codes that proteins often have, right? Because this one just told it to get to the ER. Now, if that protein's supposed to stay in the ER, there'll be another amino acid signal on it that says this is an ER retention protein. And so the ER will know to not pass that protein along to the Golgi. There's sometimes Golgi retention sequences. So a protein might get modified in the ER, it'll get butted off into a vesicle, it'll fuse with the cis side of the Golgi, but then maybe that's the protein that's supposed to stay in the cis side of the Golgi. There'll be a specific amino acid sequence that says, I'm the kind of protein that has to stay in the Golgi, and it won't get passed on. There's secretory signals as well that say, I'm the kind of protein that needs to pass through the Golgi and the ER, but then gets shuttled out to the plasma membrane and get dumped out. So most amino acids have very specific signal sequences in them that kind of guide their direction of where they're supposed to end up in the cell, so that they're at the appropriate location to do their function. Yeah.
I wonder, how does the signal recognition particle know to take it there? Like you said, I get what you're saying.
Yeah.
Kind of just tongue in cheek. It's supposed to go there.
It knows, yeah.
It knows. Is there an easier or relatively short answer to that question?
Well, this actually is a good segue into what we're going to talk about, because there's information here, right? And so I say it knows, and I'm kind of personifying it as if it's thinking or something. But there's an agreed upon signal, right? There's a amino acid signal there, and that amino acid signal is specifically recognized by this protein, this little bridge-shaped protein, the signal recognition particle. And when that guy binds to it, he can also bind to the docking proteins. So if this guy binds to the signal peptide, there's actually conformational changes in the protein that makes him capable of now docking to his docking protein. So that's what I say by no, is that that's its specific task and its specific binding affinity is for those things, right? So these signal recognition particles are floating around the cytoplasm, and when they see that sequence, that signal sequence, they bind to it, and it's a chemical interaction, right? It's an affinity. And it's got a certain amount of adhesion properties that it wants to grab that. And then now it's got a conformational change in it, and now it wants to dock to this receptor protein, right? So they don't dock without having a signal peptide stuck to them, right? They only dock when they've grabbed a protein that's supposed to go there, right? And then that causes a conformational change that swaps the signal peptide onto this other transmembrane. So we're talking about chemical affinities. And so in that sense, we're just talking chemistry. But when I say things know, it's because something or somehow has agreed that that's what should happen, right? Because that's the purpose. And I'm using all this purposeful language, and I think there is a purpose. I think there is somebody who designed things to do it this way, right? So I intentionally and unapologetically use purposefulness in my talk. But there's chemistry involved as well. But what I'm saying is the fact that it knows is that somebody set up the chemistry to do the thing you want it to do, right? You can set up a circuit, and just by the laws of physics, that circuit will transfer things through capacitors and resistors and whatever. And you can explain the circuit all by just the chemistry and the physics of circuits, right? But a designer set up the circuit that way. So I'm saying that there was a designer that set up this up, and now the chemical properties do what it was designed to do. So it's a machine, right? It's a functional machine that's been set up.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.